Hello, my name is Jenan Shareef, and I'm a junior here at Michigan State.
My name is Skedel, and I'm a sophomore. We are undergraduate students working in the laboratory of Dr. Jack Harkema in the Department of Pathobiology and Diagnostic Investigation. The title of our presentation today is Dietary Prednisone Quells Some but Not All Silica-Triggered Manifestations of Autoimmune Disease in Lupus-Prone Mice. This is work that we have contributed to in the past year.
This slide provides some key information regarding the rationale and relevance for our preclinical study. Systemic lupus erythematosus is an autoimmune disease with recurrent flares that are triggered by environmental exposures. Respirable crystalline silica dust is an occupational hazard to workers in the mining, fracking, and other stone cutting industries. When silica is inhaled, it incites lung injury, inflammation, and autoimmunity in workers genetically prone to autoimmune diseases like lupus. Prednisone, a corticosteroid, is a common anti-inflammatory treatment for lupus flaring. Prednisone's effectiveness to prevent or treat crystalline silica-triggered pathologic manifestations of autoimmune disease in the lung and kidney is not yet known.
Previous preclinical studies in our lab have demonstrated that human equivalent doses of dietary omega-3 fatty acid prevent silica-triggered autoimmune disease in mice that are genetically prone to developing lupus. In this mouse model, animals are intranasally instilled with silica. This causes the death of alveolar macrophages in the lung, releasing autoantigens, formation of ectopic lymphoid structures around airways and blood vessels in the lung, and production of autoantibodies from plasma cells. Complexes of autoantigens and autoantibodies enter the systemic circulation and deposit in the kidneys, causing injury, inflammation, renal failure, and eventually animal death. We have found that omega-3 fatty acids block this autoimmune disease triggered by inhaled silica. In the present study, we ask how dietary prednisone impacts this animal model.
Therefore, the purpose of this study was to test the hypothesis that prednisone-amended diets prevent silica-triggered autoimmune disease in lupus-prone mice.
This slide illustrates our experimental design for this study. There were 5 groups, 2 control and 3 treatment groups, with 8 mice per group. After an acclimation period at 6 weeks of age, the mice were started on either control diet, low prednisone, medium prednisone, or high prednisone diet. Once a week for the next 4 weeks, they were intranasally instilled with either saline vehicle alone or saline containing 1 milligram of silica. Their health, food intake, and body weight were monitored for the next 14 weeks until the scheduled necropsy day. During the necropsy, bronchoalveolar lavage fluid, lung tissues, and renal tissues were collected and processed for light microscopy and immunohistochemistry.
There were 4 diets involved in this study: the control diet with no prednisone, a low human equivalent dose prednisone-enriched diet which had 5 part per million, a medium dose enriched prednisone diet which had 15 ppm, and a high prednisone dose enriched diet which had 50 ppm. Unfortunately, the animals given the high prednisone-enriched diet developed steroid-induced toxicity and had to be humanely euthanized prior to the scheduled necropsy day.
This slide shows our histopathology methods performed on the lung tissue taken at necropsy. Lungs were inflation fixed with a formalin fixative. After fixation, the left lung lobe was sectioned in 4 for histochemical staining with hematoxylin and eosin for microscopic identification of histopathology. Immunohistochemical staining was used to identify B and T lymphoid cells and plasma cells in the lung. Standard morphometric methods were used to measure lymphoid cells from digital images of lung tissue. Reference tissue, which includes both hematoxylin and IHC stained tissues, was counted using a 4-point grid. This value gives the area of the total stained tissue. Then, for each lymphoid cell type, we counted only the cells stained positively with red chromogen that intersected with a 144-point grid. After dividing the positively stained points by the total tissue points and multiplying by 100, we got the percentage of positively stained area. This correlates with the density of the different lymphoid cells, as you will see in later results.
Light photomicrographs of lung sections stained with hematoxylin and eosin were used to identify histopathology, including ectopic lymphoid tissue in the lung. The top left image is photolight micrographs of the control animals with no prednisone, where PV stands for pulmonary vein, PA stands for pulmonary artery, BA stands for bronchiolar airway, and AP stands for alveolar parenchyma. The image to the right of the vehicle control is the silica control group, where you can see a dramatic influx of ectopic lymphoid tissue surrounding the airways and blood vessels. Here is the low prednisone group with a visible attenuation of ectopic lymphoid structure, and to the right is the medium prednisone dose with an even larger difference. The graph on the right shows a semi-quantitative analysis rating the severity of the ectopic lymphoid tissue in the lung of the vehicle control, silica control, low prednisone, and medium prednisone dose groups. This graph illustrates that low and medium dose prednisone attenuated silica-triggered ectopic lymphoid structure formation in the lung.
Immunohistochemical staining for CD45R positive B-cell infiltration in the perivascular tissue in the lung was then performed. The top left image shows the vehicle control group with the tissue surrounding the blood vessel. E stands for endothelium, IS for interstitial space, and A for alveoli. As seen in these two images at the bottom, there is significantly less B-cell infiltration in the low and medium prednisone dose groups. The morphometric analysis determined that there was a dramatic influx of B cells in the silica control group compared to the vehicle control group. In addition, the medium prednisone group significantly impeded B cell infiltration when compared to the silica control group. As seen in the graph on the right, prednisone attenuated silica-triggered CD45R positive B cell infiltration in both low and medium doses.
Next, T lymphoid cell infiltration in the lung was marked using CD3 staining and analyzed. As you can see, the percent of CD3 positive cells, colored red, had a different response than the CD45R stain. Per the graph on the right, there was a significant increase between the vehicle control group and the silica control group. There was, however, no significant difference between the silica control group and any of the prednisone treatment groups, showing that prednisone did not impede silica-triggered T cell infiltration in the lung.
Next, I counted the plasma cells within the lungs using IgG staining. In this case, the positive brown stained cells are plasma cells surrounding a blood vessel. These graphs show our morphometric analysis. Although the only significant difference in IgG positive plasma cell percentage was between the vehicle control and silica control groups, the medium prednisone dose modestly attenuated silica-triggered IgG producing plasma cells in the lung.
To determine the abundance of IgG autoantibodies found in the bronchoalveolar lavage fluid, samples were sent to our collaborator, Dr. Lee, at the University of Texas Southwestern. There, Dr. Lee conducted a microarray analysis of the various autoantibodies found in the BALF. These were then categorized and graphed. This graph shows the abundance of the total IgG autoantibodies in the BALF. The data shows a significant difference between the control groups as well as between the silica control and the medium prednisone dose treatment group, showing that the medium prednisone diet only modestly attenuated the IgG autoantibodies in BALF of the lung.
We also examined the kidneys using light photomicrographs and immunohistochemistry. The semi-quantitative severity score analysis illustrates that medium prednisone dose significantly attenuated but did not fully impede the silica-triggered glomerulonephritis. Between the silica control group and the vehicle control group, there is a significant increase in glomerular size and increased thickened membrane, as well as increased deposition of proteinaceous material in the renal tubules. As seen in the images, the sizes of and thickening of membranes within the glomeruli are visibly attenuated in the medium prednisone group compared to the silica control group.
Finally, our results conclude that the high prednisone diet caused marked toxicity and the medium prednisone diet caused mild toxicity. The medium prednisone diet significantly reduced pulmonary CD45R positive B cell infiltration and some IgG autoantibodies in the lung. The medium prednisone diet did not ameliorate silica-triggered CD3 positive T cells and only modestly impeded IgG producing plasma cell infiltration in the lung and glomerulonephritis in the kidney. In conclusion, human equivalent doses of dietary prednisone quell some, but not all, silica-triggered manifestations of autoimmune disease in lupus Crohn mice. More effective and non-toxic treatments are needed to prevent toxicant triggers of autoimmune diseases in occupational settings.
Finally, we would like to acknowledge and thank all those who have contributed to this project.